And we are good there. So we'll get started. So today is a very exciting presentation from a ship off the coast of Puerto Rico. That is so cool. And our speaker will be introduced by one of our students. But before she does, I just wanted to say too, that the department that he's representing at URI is actually one of the world's premier institutions for oceanography. And I believe my interest in looking you up was you found some new chemistry involving oxygen at the seafloor level or something. That was how I was like, oh, wow. So anyway, that's a little bit of background. Our introducer, in fact, our co-host today is Yishan Jin, and she is originally from China. She is a biochemistry major here in our department. She is very international. She went to high school at a Seventh-day Adventist high school in Thailand, and it's called Trinity International High School. She's planning to get into dental school after graduation. And one of the fun facts about her is that she can memorize 10 digits or more of pi. She told me right now she could probably only do 10, but she's done more than 10 in the past. So Yishan, you want to unmute and go ahead and introduce our speaker.
Thank you, Dr. Marie. Today, it's my pleasure to introduce you. The speaker of this day is Dr. Stephen Dong. Yeah. Dr. Stephen is actually a professor of oceanography at University of Rhode Island. And his research is focusing on the interaction between biosphere and the physical world. The interesting about the microbe in the subsea floors is that they have extremely slow metabolic rate, which make them persist in sediment for at least 100 million years. Another thing about them is that they breathe up 1 million times slower than the microbe in the surface world. They also control Earth's surface oxidation and major aspect of ocean chemistry. They affect climate on long time scale, and they also play major role in forming and destroying economy resources. So let's welcome Dr. Stephen.
Very good. Thank you.
Much. So I'm going to, it's a pleasure to be invited. I'm really glad to see all of you here today from both Andrews University and from I'm delighted to see people here from the West Indies today. This is, to me, this is a huge advantage of, these Zoom seminars, as you can participate from throughout the world. Thank you all for joining, and thank you for inviting me. So I'm going to speak today about life beneath the seafloor, as we heard. And I've somehow frozen my slides, so let me, I'm going to stop sharing and then start sharing again and see what happens. Okay. There we go. So the primary research subject I'm going to discuss today are microbial organisms that live beneath the seafloor. And microbial life, of course, is in the world around us. There are microbes living in the atmosphere, microbes living, on the land, in the soil, microbes living in our digestive tracts, on our skin. Everywhere in the world there are bacteria, and in most places in the world there are archaea, which are another kind of microbe. And then, of course, little eukaryotic microbes, like amoebae. But the main ones we see beneath the seafloor, I'll get into in a minute, are bacteria and archaea. So this is our primary research subject, is shown here as a little glowing green dot. And it glows green because we add a dye to the sediment or to the samples that binds to nucleic acid. And then through a microscope, that dye fluoresces green. In this case, it's a dye called cybergreen, so it fluoresces green. Other dyes fluoresce red or yellow or orange. The platforms that we use to recover these microbes, I've put 3 pictures above. The one on the far left is a US leased drilling vessel that's owned by a Norwegian ship handling company. And the primary funder of that is the US National Science Foundation. I've used that ship for a total of about 6 months in 2-month increments over the last few years. And the ship in the middle is the ship that I'm on right now. It's the research vessel Neil Armstrong, which is operated by Woods Hole Oceanographic Institution. It's part of the US National Research Fleet. The research fleet is scattered throughout different institutions. So we have a ship at the University of Rhode Island. There are a couple of ships at Woods Hole. There are a couple of ships at Scripps. There's a ship at Oregon State, a ship at the University of Washington. There's a ship in the Gulf Coast that's operated by a consortium of states. And there's about 18 ships and boats of different sizes in the National Research Fleet. This is it's kind of a mid-sized ship. You know, you can think of this as kind of the Honda Accord of oceanographic vessels. It's very new, but it's not particularly big. It's about 230 feet long. And for example, the ship I spoke of a minute ago, the Norwegian-owned one that the US leases, is 471 feet long. And then the ship on the far right is the Japanese drilling vessel Chikyu, which is 560 feet long. So the Chikyu is capable of drilling kilometers beneath the seafloor in water shallower than 4 kilometers. The Resolution, which is the one on the far left, is capable of drilling in up to 6 or 7 kilometers of water. But it's got a different kind of rig. I'm not going to get into the details. The one on the Japanese ship is more like a standard oil and gas rig. And the one on the US ship is a lighter rig, so it can work in deeper water with longer pipe. And then the ship I'm on right now has no drill pipe at all. We use wireline operations. We essentially put coring devices on rope and lower them to the seafloor and then core from the seafloor. So one last thing I want to say before I move on is this particular image of this little microbe, this lonely little microbe, is from beneath the North Pole. There was a drilling expedition in the early aughts at the North Pole by the international scientific community, and David Smith, who's on this ship with me right now, sailed as a microbiologist and took this photo of a microbe from about 230 meters beneath the seafloor at the North Pole. So even up there, there's microbes in the sediment, beneath the ice and beneath the water. This is an image from a paper by one of my postdocs. It's now a decade old. And in it, we to make this image, we counted cells in marine sediment and then calculated from that the number of cells per cubic centimeter of sediment, and then calculated from that the number of cells at each site. And that gives us the color within the dots. And then we correlated those total cell numbers to properties of the surface world, like distance from shore and chlorophyll content of the overlying water. And we then use those correlations to project throughout the rest of the ocean to create the color fields that you see. And so what you'll see is that there's generally a pretty good match. The dark blue circles of ours tend to be near the dark blue fields that we project. The green circles tend to be in the green fields that we project. The red circles tend to be in the red fields that we project. And so the similarity dots in the field around them gives you a sense of sort of the robustness of our projection. And then you'll see spots like where we'll have a green spot in an orange field or a red field or a blue spot in a green field. And that tells you we didn't do such a good job of projecting there. It turns out that our unexplained variation, what we call the residuals, are evenly distributed about the mean of our projections. And so that tells us that even though we're not great at every site, we're pretty good for the ocean overall. So then we can take the sum of everything we project in all of those spots and total it up, and we get 3 times 10 to the 29 cells per cubic, or in all the sediment of the world ocean. So that's essentially, 3 with 29 zeros after it. And that's roughly equal to the total cell number estimated for the ocean. And that's roughly equal to the total cell number estimated for soil in the world. But it's a sense of Earth's total biomass, because the cells we see beneath the seafloor are really tiny. And they're really tiny for reasons that I'll explain as I go through this presentation. So there's 3 big categories of life on Earth. You probably all know this from your biology classes, but there are bacteria, there are archaea, and there are eukarya. And we categorize them, and we put them into categories based on, they're not really based on similarity in the total DNA sense. They're based on shared unique reads, shared unique pieces of DNA. So if you look at this plot, for example, animals, fungi, and plants have DNA sequences in common with each other that they don't have with anything else. Or if we shift over to the bacteria and look at that, we'll see that proteobacteria and spirochetes and cyanobacteria have reads in common with each other that they don't have with Thermophex or Aquatoga or any archaea or any eukarya. And so that's the standard tree of life now that genetics experts have put together over the last, since basically the mid-70s. So all 3 kinds of life live actually deep beneath the seafloor. We have lots of bacteria down there, we have lots of archaea down there, and we have very few kinds of eukarya down there. Basically, the eukarya that we know are down there are limited to fungi. So essentially mold and mushrooms. And then we have archaea and bacteria. So this is a plot that shows us how sort of bacterial diversity varies as you go from the sea surface to beneath the seafloor. And when you squint at this plot, on the far left, I have something called OTUs. Those are operational taxonomic units. You don't have to really worry about what the operational properties are, although I'd be happy to describe it if someone asks. But essentially, you can think of each OTU as sort of like a species of bacteria. So there's a number of kinds of bacteria that we have, and these are color-coded. So all of the red colors are from the same site, all of the green colors are from the same site, all of the blue colors are from the same site. And what you see is that we have our greatest number of OTUs, so our greatest richness in types of bacteria, in the O2 minimum layer, which is an oxygen pore layer beneath the surface of the ocean. And then we have roughly the same number
Looks like we're experiencing a delay or something.
Yeah, he is on a ship.
Yeah, yep. See if he comes back. He might be coming back in. Yeah. I think he's trying to re-enter or something. Yeah, we worked on this yesterday. There was a significant delay at times. Today it's working okay, except now.
Some good Henry's Law questions there on that oxygen concentration.
Huh?
there's some Henry's law questions on
Questions. Oh, yeah.
the oxygen as a function of depth
Of depth.
graph.
Yes, that's right. Yep. That's right. Let me see if I could connect with him by email. I don't have his number. I'm pretty sure he's trying to get back on. Okay. This didn't happen yesterday. I do have his backup slides, but that's not going to do us any good because I don't I don't know the topic. So I just sent him an email to see if he responds and gives us some kind of feedback. So this is what happens in real life. Out on a ship in Puerto Rico, off the coast. Yep.
So I actually have a question that is not a question that shows I do not know anything about biology, but about that tree of life. Dr. Murray, did you have his slides?
I do have his slides. I could go try to find them now.
I took a screenshot, so if you want to just
Uh-huh.
make me a co-host, that works too.
Okay, we could do that.
I'm just filling time. If he comes back, the right thing to do is give him the floor.
Yeah, so you're a co-host now.
Super.
Okay, he's coming back in. I just
Perfect.
admitted him back in.
Never mind then.
So you had a question on the tree of life, huh?
Let's let him continue.
Okay. There he is.
I'm very sorry. The ship server cut us off because it said I'd exceeded my daily allocation and they forgot to turn that off. And they've promised me that won't happen again, but I'm very sorry.
Oh, it's all right. We totally understand.
I appreciate your patience, but
Oh, no problem. We've actually gained more people since you left.
Okay, that's good. So I'm going to go ahead and share screen again.
Okay.
Oh, yeah, I need you to set me up as a co-host
Set you up
again.
as a co-host again. Yep.
Yeah.
There you go. There you go.
Thank you.
Very nice.
And
So you were
We
talking
will be right back
about
there.
slide 5, I believe, is where we left off.
Right.
Yeah.
So the basic point is just that diversity is lower in the sediment beneath the seafloor than anywhere above in the ocean.
Mm-hmm.
Having said that, this is from a paper that I published with some Japanese collaborators. I didn't change this. It says Impress, but it was published last year, 2021. And because the sediment includes the seafloor as well as the sediment below, it turns out that if we project from our known number of kinds of organisms in all of these environments, we have roughly the same number of kinds of bacteria and archaea total in marine sediment as in topsoil and in the ocean. So just as the total cell numbers are about the same, the microbial richness, the diversity is about the same. And they have fairly big effects because the organisms that live beneath the seafloor, particularly in the sediment, traditionally their main food source is thought to be buried organic matter. Although I will challenge that a little later in my presentation. But their consumption of buried organic matter changes the chemistry of the ocean. And so we get burial of organic carbon and organic nitrogen, just organic matter that's created by photosynthesis in the world above, falls to the bottom and gets buried with the sediment. Organisms that live beneath the seafloor use a variety of oxidants to eat that. The ones that live near the seafloor or deep beneath the seafloor in the open ocean use oxygen and to eat their organic matter, just like we do. Other organisms use nitrate as their oxidant or their terminal electron acceptor. Others use sulfate as their oxidant or their terminal electron acceptor. Others use oxidized metal that's buried in the sediment, like iron oxide, manganese oxide. And those of you who are, you're all chemists, or many of you are chemists, so you're aware that the manganese and the iron, and for that matter, the sulfur, they don't need to be bound to oxygen to be oxidized, right? They just need to be able to take electrons, so they need to be in a reduced form. But a lot of the iron and manganese and other metals initially buried in the sediment are in an oxidized form, and then that gets reduced by microbial activity coupled to the oxidation of organic matter. So organisms are down there eating some fraction of the organic matter, converting it to oxidized carbon. The ones that are reducing sulfate are removing negative charge from the ocean because the sulfate's diffusing down from above. And the ones that are reducing nitrate are also removing negative charge from the ocean. It's the nitrate's diffusing down from above. But there's a lot more sulfate than nitrate in the ocean, so the sulfate respiration is a bigger effect. Essentially what that means then is they're pulling oxidizing power out of the ocean and using it to oxidize sediment. Some of that oxidizing power comes back to dissolve CO2, and that dissolved CO2 takes up hydrogen and comes back out in as either HCO3 or CO3 2-, which in oceanographic context we call alkalinity. And what that basically means is that the charge that's removed by reducing the sulfate and precipitating iron sulfide, much of that comes back it basically all comes back in the form of alkalinity. Essentially oxidized CO2 that is shifted around with water to get the charge balance in the ocean back. So what we see here then is that what this cartoon shows us, and if anyone has any questions, feel free to ask. What the cartoon is showing us, this is a major sink for sulfate from the world ocean. In fact, it's the dominant sink of sulfate from the world ocean. It's the dominant sink of nitrate from the world ocean. It's a source of dinitrogen to the oceans and atmosphere. It's a source of alkalinity to the ocean. It's probably the primary source of alkalinity to the ocean. The burial of that organic matter and the iron sulfide that gets produced when the sulfate is reduced is a sink for reducing power from the surface world. And so in a sense, you can think of that burial of organic matter as what allows the world around us to be oxidized. And the microbes that live in the sediment are the last filter on the escape of that reducing power. So they're busy reoxidizing organic matter and releasing it back into the sediment, where some of it diffuses up. That oxidizing power diffuses up and comes back out, is essentially in the form of carbon oxides. So big global effects in the long run on the alkalinity and on the chemistry of the ocean and atmosphere. And this is overriding my slides again, so I'll end it and start it up again. And we'll go right back to where we were. I'll actually go to the next one. And these are essentially a table showing the same thing. It's the primary, that burial of organic matter is the primary flux of carbon and nitrogen from the surface world. That reduction of sulfate in the sediment is almost the entire flux of sulfur from the world ocean. The burial of that organic matter is also the primary flux of reducing power from the surface world. It's partially the oxidation of buried organic matter gives us what may be the primary flux of alkalinity or charge back into the ocean. And that nitrate reduction is the primary sink of bioavailable nitrogen from the world. So you can think of it as removing fertilizer, in a sense, from the surface ocean. Despite their big global effect, these microbes breathe very slowly. So we've essentially done mass balance calculations to see how much sulfate reduction is going on, how much oxidized carbon production is going on. So this cartoon here shows you the amount of CO2 that's oxidized by the average cell in a pure culture of sulfate reducers on a bench in Denmark. It shows you the amount of carbon that's oxidized per cell in shallow sediment on the coast of Denmark. I just got these data from Danish colleagues. And then the next two narrow bars show us the amount that we infer from the sulfate reduction rate, because each sulfate can be used to oxidize 2 organic carbons. And then the amount that we infer directly from the production of carbon, oxidized carbon from CO2. And those two numbers agree fairly closely, which tells us that the net oxidation of organic matter is probably pretty well constrained in these hundreds of meters of marine sediment represented here, and that the main oxidant is sulfate. But the big takeaway you should get from this slide is that the average microbe in sub-seafloor sediment breathing up to 6 orders of magnitude more slowly than the average microbe on that Danish lab bench. And they're breathing about 3 orders of magnitude more slowly than the average microbe in that seafloor of the Danish estuary. So these microbes on average are breathing very, very slowly. Now, we don't know how fast individual microbes are breathing. It could be that 1 in a million is breathing as fast as the microbes on that Danish bench, and that the other 999 aren't breathing at all. We don't think that's really happening. But the point I'm trying to make is that the average can be misleading. Some may be breathing much faster, some may be breathing much slower. So at this respiration rate, it takes hundreds to thousands of years for the average microbe down there to produce enough biomass to replace the molecules in its body. They're just breathing ridiculously slowly. So either we're missing a critical source of energy, or these things are living fabulously slowly. This is stopped again. But before I dive into the next topic, I want to make a point, which is that these things can't be reproducing faster on average than they're replacing their biomass on average. So this means that they're on average living for hundreds to thousands of years. And they could be living much longer because, they need to do sort of maintenance molecular replacement as well as creating biomass to reproduce. The next topic I want to talk about, and so what you're going to see is every little module I present to you today is going to end with a mystery of some kind. The mystery there is, how long are these things really living? And the answer is, we don't know. They're living a long time, but we don't know how long they're really living. This next one, I'm going to talk about life after being trapped since the age of dinosaurs. This is from a study with some other collaborators, mostly Japanese, that was published 2 years ago. Essentially, my collaborator Yuki Morono took little bits of sediment from an expedition we did in the South Pacific, and he gave them different food sources. So he gave them ammonium, he gave some nitrate, which isn't really a food source, but an oxidant. He gave some of them mixed amino acids, he gave others specific amino acids. And those sources of food or electron donors and those oxidants like nitrate were isotopically labeled with heavy stable isotopes. So he'd add carbon-13 to the amino acids before the amino acids were given to the sediment. And then we would wait to see how long it took for the microbes to incorporate the carbon-13 into their bodies, or to incorporate the heavy nitrogen into their bodies. And then he would use a technique called nanosims to image that. Hey, Evan, I hate to do this because it's a public space, but I'm giving the seminar right now. So if you guys could talk in the lab. Thank you. I wanted to clear down. I didn't know if you guys could hear that, but I wanted to clear down the background noise a little bit. Okay, so the green glow is DNA, that's cyber green again. The blue glow, the nanosims technique shows us preferentially where the heavy isotope of carbon has been taken up. And the warm glow shows us preferentially where the heavy uptake of nitrogen has been taken up. And just so you can convince yourself those are actual cells, there's an image of the exact same naked cells on the far right here. So what we see is that this particular sediment was 104 million years old. The microbes that came out of it have DNA, so they glow green. They take up, given enough time, the heavy carbon, so they glow blue, and they take up the heavy nitrogen, so they're glowing orange with these filter sets. So what that basically is telling us that even though these things have been starving for 104 million years, if we offer them a buffet, they wake up and they eat it, and they incorporate it into their bodies. And this next slide shows us that they not only do that, they reproduce. So if you look at these plots, these are for 4 different samples of different ages, but all, one's 13 million years, one's 4 million years, one is 102, and one is 95. So the starting point for the number of cells per cubic centimeter in all of these little experiments is the gray dot on the far left of each plot. And you see that after several tens of days, they have increased their abundance by 3 to 4 orders of magnitude. So they're actually partying. They're not just eating that material and sitting there and incorporating it into their bodies. They're eating that material, they're incorporating it into their bodies, and they're multiplying. And they continue to maintain high concentrations for pretty close to a couple of years, at which point the experimental material was exhausted and we stopped them. We stopped the experiments. So that tells us that even microbes from the age of dinosaurs, from marine sediment, can be sort of coaxed into normal living activities, into eating and incorporating what they eat into their bodies and into multiplying. So the last thing I want to talk about is perhaps the strangest thing. I'm going to tell you that we have some evidence that life beneath the seafloor is sustained by chemicals from radioactive splitting of water. So in a sense, what I'm telling you is, in fact, those organic matter-based calculations are not complete, that we're missing an energy source. And that energy source is providing, depending on the site, up to about 10 times more energy than the organic matter oxidation. So let's take a look. And I probably shouldn't phrase it in terms of energy, but if you think in terms of electron fluxes, there's about up to about 10 times as many electrons accessible to these organisms for redox activity from radioactive splitting of water is from oxidizing buried organic matter. So the first point I want to make is that splitting water by natural radioactivity produces hydrogen and oxidants. This process was first discovered in Madame Curie's laboratory in 1906. A man named de Bairn did some experiments and found out he was getting hydrogen when he irradiated water. And I'm phrasing it a little differently. It was more of an accident than that. He started noticing he was getting buildup of hydrogen in one of his experiments, and he inferred that it was from the water in the air of his experiments. And then he set out to do some tests, found out it appeared he was correct. And then he did some more experiments, and he found out that the hydrogen was being balanced by production of hydrogen peroxide. And so if he took the amount of hydrogen peroxide he got and the amount of hydrogen he got, he could account for the missing water. And he'd done that by 1909. They were very elegant experiments. And this has been an issue ever since in nuclear science and engineering. There's actually, and I'll just say this conversationally, there's actually about 30 different products of the splitting of water by natural radioactivity. And some of them are truly extraordinary for me. I've got a fairly elementary chemistry background. Some of you you all have a much better background, but there are things like chargeless electrons created, in this process. And then a lot of the really exotic stuff collapses back into a more normal state in something like 10 to the minus 12 seconds. So by 10 to the minus 9 seconds, we have atomic configurations we're more used to. And then shortly thereafter, we're down to basically dihydrogen and hydrogen peroxide and water and some oxygen, depending on the chemistry of the environment. So this hydrogen and the oxidant can, in principle, fuel subsurface life. In the late 1800s, German scientists discovered these things that they called canal gas bacteria, which means boom gas in German. And what they call them that because dihydrogen is of course explosive. And so that is boom gas bacteria. They take the boom gas and they use it to make water, and they get energy from that. So in one sense, what I'm about to tell you is really coupling two ideas that are like more than 100 years old, but we're doing it in a somewhat different context. So essentially, in the subseafloor, we think there are two different sort of generic metabolisms. There's the activity that's fueled by the surface photosynthetic world, where organic matter is produced in the surface world, sinks to the seafloor, and it gets buried. And then the microbes that live in the sediment use the oxidants around them, oxidants that are either buried with them or that diffuse down from the ocean above or up from the aquifer and the basalt below. And they use those oxidants to essentially oxidize the buried organic matter. And you can transfer 4 electrons per mole of carbon in this reaction and create CO2 and water. The other pathway is the one that's fueled by this radioactive splitting of water. And in that scenario, splitting water gives us hydrogen and various oxidants, which I've here just represented with 1/2 O2. And the organisms take those products and they put them back together to make water again. The reality of the system in both cases is more complicated because organisms can also reduce and oxidize intermediates in this net pathway, right? So something can reduce sulfate to sulfite and then S0, and something else can disproportionate S0 to make sulfite and HS minus. So there's a number of ways that this reducing power or oxidizing power can be moved about through the ecosystem, but the net reactions are the ones that I've shown here. And here's where it gets kind of not very 100-year-old. We've been doing experiments in my laboratory when I'm not at sea. Most of these experiments were done by my student, Justine Sauvage, who's now a postdoc in Belgium. But we used polonium as an alpha source and cesium as a gamma source, and looked at how much hydrogen is created in pure water, in seawater, and in wet marine sediment of different types. There's a map there that corresponds to the color bars in those plots and that tells you where the sediment came from. And what we find is that pure water and seawater give us about the same amount of hydrogen per unit of radiation energy. So 100 electron volts is what we're normalizing it to here. But then you look at the colored bars below, and you see that pretty much all of the sediment, the wet sediment, yields more hydrogen for the same amount of radiation energy. So the sediment is catalyzing the production of hydrogen from radioactive splitting of water. That catalysis is much stronger for alpha radiation than for gamma radiation, if you look at the units at the top. And it's much stronger for abyssal clay than for other types of marine sediment. So the clay is generating a lot more wet clay is generating a lot more hydrogen than wet silicious ooze, which is mostly wet opal, or wet marl, which is mostly wet calcium carbonate. So in a sense, the sediment is acting like an LED or a semiconductor. And the cause of this catalysis is not, as far as I know, accepted fully. I certainly don't contribute to the theory directly. There's a couple of different theories out there. One theory is that the minerals scavenge the intermediate products of the radioactive degradation of the water. That theory is almost certainly not true, because there's not enough time for all those products to diffuse back together again, you know, over the path of the cross-sectional path of, say, an alpha particle. The other theory, which comes out of the nuclear science industry and experiments with uranium dioxide that other people have done, is that the sediment, well, they're talking about uranium dioxide, they're not talking about sediment, but their theory is that the oxide literally acts like a semiconductor, and excitons are created within the solid phase and propagate to the surface of the material, where they split electrons across, or split water across a broader front and multiply the yield. I'd welcome thoughts from any of you at the end of this, if you have, you know, about this. Whatever the cause is, you know, our experimental result is reproducible. We see it over and over and over again. And with every run of mineral experiments, we do pure water experiments as a control. So, you know, with every run, we can normalize our results to the pure water. And we see that the wet sediment multiplies the production of hydrogen over and over again. Perhaps the simplest scenario that might explain all of this is that the catalytic minerals are zeolites, which some of you probably know are active in a lot of weird chemical transformations. And zeolites are perhaps the most abundant mineral type in the abyssal clay. Although they're called clay, they're really mostly zeolite. And what we see in the other categories of sediment, like the silicious ooze, the marl, the calcareous ooze, might simply be dilution of zeolite by the calcite or the opal or something else. With that, I'll move on. So this plot shows the ratio of radiolytic hydrogen consumption to organic consumption in electron equivalence. So we just say, okay, for each carbon that's oxidized, we can move 4 electrons. For each hydrogen that's oxidized, we can move 2 electrons. And our consumption of hydrogen is equal to the production that we calculate based on those experimental results. Because when we measure hydrogen in the sediment, it's always below detection. And it should be orders of magnitude above detection if something's not consuming it, at least in the middle of the column, given the timescales of diffusion. So what we see when we plot the ratio of these two things, the organic consumption that we calculate from, say, the production of oxidized carbon dissolved in the water, and the hydrogen consumption that we calculate from the experimental yields multiplied by the thickness of the column and subtracting essentially the missing hydrogen, is that in sediment that's younger than a few million years, the predominant food source, or the predominant redox activity for the microbes, is oxidation of organic matter. But once the sediment's older than a few million years, there's much more hydrogen disappearing than there is CO2 being produced. And so we think that once the sediment's older than a few million years, and it's buried more deeply and farther from the ocean, that's the source of oxidants diffusing in for the net process, radiolytic hydrogen takes over as the primary fuel for the subsurface life. And you see there's a transition point that varies from site to site, but it's generally a few million years down in the age of the sediment. And one other thing I'm going to say, and this gets back to this issue of a cascade. When you break up water in a mineralogically diverse environment, you're breaking up water in a chemically diverse environment. So there's all kinds of reduced iron around, all kinds of other reduced metals around, there's reduced sulfur around. And you're going to generate a whole, when you've got hydrogen peroxide or weirdly charged states of oxygen, you have the capacity to oxidize much more than just hydrogen. And so we think that the oxidants that are produced through this process are probably being used to oxidize a wide range of things, that they're not simply going back to oxygen or hydrogen peroxide. And if they are, then that in turn is being used to oxidize other things fairly quickly. And when we look at the transcriptomes, which are essentially the expression of RNA in the microbial communities that we take from the sediment, we see some evidence for that. We see lots of genes that are used in neutralization of reactive oxygen states are expressed on the far right of this plot. We see genes that are involved in sulfur and iron oxidation are expressed heavily. So the size of these circles represents how heavily expressed they are. We see that there are genes associated with reductionate and selenate and nitrate, which shouldn't be there deep beneath the seafloor unless something's creating them, because they should have all been reduced in shallower sediment. And I'm not showing you the genes for hydrogenase expression because they're just ubiquitous. So you need hydrogenase to use the dihydrogen, and every sample has lots of hydrogenase expression. We've actually developed a proxy for hydrogenase activity using tritiated hydrogen to check this, and we see hydrogenase expression in all marine sediment. So that was left off of this plot, but I wanted to show you that there's a diverse range of oxidized species being used. And this is in sediment where, according to sort of traditional theory, the canonical theory, the only thing available to be reduced should be CO2. But there's lots and lots of different species being reduced, which probably tells us there's lots and lots of different chemical species being oxidized. So I want to sort of swing to a last point, which is that if this process can sustain microbes beneath the seafloor, it can potentially also sustain microbes on other worlds, on early Earth, prior to the advent of photosynthesis, or on worlds like Mars, or moon worlds with icy moon worlds like Europa. So in summary, life beneath the seafloor is abundant and diverse. It respires very, very slowly. It can persist for 100 million years and then return to normal. And we think it's probably principally sustained in sediment older than a few million years by radioactive splitting of water from natural decay of uranium, thorium, and potassium. Finally, it serves as a model for life on other worlds. The last thing I want to say is there's lots that we don't know, some of which we're sailing right now to test. We don't know how they serve, how they adapt, and how they evolve on so little energy. We don't know how they interact with each other. We don't know how long individual cells live beyond the hundreds to thousands of years that it takes to turn over their biomass. And we don't know their ultimate limits in depth and energy. So we know they're living very, very slowly, but for all we know, cells can live even more slowly. And we don't know how deeply they live beneath the surface of Earth because we haven't gotten to the bottom yet. We know that where we've got really hot sediment or really hot water coming out of the ocean, there's no life at great temperatures, say above 120 or 130 degrees Celsius. But in cooler sediment, it's low in energy. We have no idea how far life goes yet. And with that, I will stop and open up for questions, comments.
Okay. Well, thank you very much. That leaves a lot of interesting questions. I'm just like scratching my head about some of these things. For example, I didn't know anything about radiolysis of water. And the historical precedence and everything for that. That itself is pretty interesting, that it's something that's known for 2 centuries. Is it when was
Well, roughly, yeah, a little, I guess 115 years.
yeah. So it do you
Go ahead.
do you know of other maybe physical chemists that maybe sort of piggybacking on this and trying to really figure out this radiolysis system?
That's a good question. I'm going to back up and say, Dick Morita and Claude Zobel back in the 1950s actually suggested that this was a source of oxygen for life in abyssal clay, but they didn't really do anything more than throw it off in a discussion. And then beginning around in the late 80s, a microbiologist in Norway named Karsten Petersen began promoting this as a process that might support life in deep continental aquifers. And then beginning around the turn of this millennium, a collective of scientists from Princeton and Toronto and Indiana University began actually looking for deep gold mines of South Africa and Canada. And they think they've got a community down there 3 or 4 kilometers beneath the seafloor in the water that the miners blast into. And I think that community is living from this process. But we're the first group to actually, take this sort of experimental approach to it and see, how much hydrogen could we get from this? And then we're also the first to really take a serious look at the mass balances to see, how much, say, electron flux might come from that relative to organic matter oxidation.
So where's the radiation?
So people have been working on it for about 30 years. Yeah.
Where's the radiation coming from? Where's the alpha particles and the gamma radiation coming from?
So the alpha particles are coming from uranium and thorium buried in the sediment.
Okay.
And, the minerals all contain uranium and thorium to some, I shouldn't say all, but many of the minerals contain uranium and thorium. Potassium is the most abundant radioactive element in the sediment, but as you know, it's basically a source of gamma radiation for us. So the thorium, there's what's called excess thorium in the first couple 100,000 years of sediment at the seafloor, because thorium's scavenged by organic matter in the ocean, and there's a daughter product of one of the uranium series that gets buried and then disappears. It's got a half-life of a few tens of thousands of years. So the main sources of radiation are the long-lived thorium and the long-lived uranium that are in the minerals that get buried.
Okay. And those are pretty widespread across the globe, correct?
Right. And so to a first approximation, the uranium and thorium content of deep sea clay is about the same as the uranium and thorium concentration of continental crust.
Okay.
So when someone shows you the mean uranium and thorium of continental crust, they're giving you about the same values that we have in deep sea clay. And then the values are lower in carbonates and opal and ooze because the minerals with more radioactive elements are diluted by the remains of the give me like 5 minutes. Yeah. The sinking microfossils.
Okay. Others have questions, I'm pretty sure. John, I see you have a question in the chat. You could, John Rosenberg, you could unmute and.
Sure. I was just wondering if I heard you talk about how as they tend to age, they lean more toward getting their energy from the splitting of hydrogen, water. So do they ever lose their aptitude for getting their energy from carbon?
So I'm going to give 3 answers. The first I'm going to say is we've never really tested this directly, but it's a great question. The second thing I'm going to say is that people have actually found bacteria living in, wastewater from nuclear power plants and in the water that's used to bathe nuclear reactors.
Mhm.
So they've found very radiation tolerant bacteria.
Mhm.
But and people have hypothesized that they may be harvesting energy from this process, but I don't know that it's demonstrated in that environment. The third thing I'm going to say is that those experiments with that 100-year-old community, 100 million year old community, suggest that they don't lose their aptitude for getting energy from carbon, because those things have been buried for 100 million years. If we're right in our hypotheses, they've been getting most of their energy from radioactive splitting of water for quite a while. But when Yuki Morono offers them an organic buffet, they take it up with enthusiasm. I can tell you that, yeah, it's resurrection.
Resurrection for
biology,
bacteria. Oh boy.
I was going to mention one more thing. There's we've begun looking at selection in these environments, looking at the genomes, and there are some properties that are less abundant in subseafloor microbes that are more abundant in the world around us. So, for example, genes for motility in some categories of bacteria are less common in the subseafloor relatives than in the relatives that swim around in the ocean. But we don't think they're evolving toward that state, because there are identical copies swimming around in the ocean. And it's misleading to say they're swimming. They just attach to particles, like sinking organic matter. So the kinds of properties that we know disappear from these communities once they're buried, it looks like what's going on is just selection for really for starvation artists. They're not really evolving new capabilities in any way.
So one other question
Go ahead, John.
Another question then was, what happens if, like, have you seen any of these any other cells that could have descended from these cells and not show the ability to get energy from the splitting of water, but be able to do that because there's, they're getting energy from the carbon. But if you took the carbon away, they would be able to get energy from the splitting of water. Like, is that something that's common in cells, like that we like all around us in these microbes, or no?
Well, so what are called chemolithotrophs are very common in the world around us. Things that get their energy from inorganic reactions, right? They might get their energy from, say, oxidizing sulfur or oxidizing metals. So it's a pretty common life strategy for bacteria to get their energy from purely chemical reactions that don't involve organic matter. The canal gas bacteria, I don't know for sure. I mean, they definitely get their energy from water. I don't know whether they have the ability to use organic matter or not. Hybrid lifestyles in bacteria, where they get their energy from an inorganic reaction, but they build their body out of what they scavenge from the environment. So you can think of them as like little Frankenstein cells. It's cheaper to take your neighbor's arm than to grow your own. That's an ugly metaphor, but that's what that's what some bacteria do.
Yeah.
So I don't think, Jonathan, I don't think, John, I fully answered your question, but we haven't pulled anything out of this sediment that exclusively lives on radiolytic hydrogen. Everything we've pulled out that we've experimented with can live on carbon. Okay. But organisms are known from the surface world that don't need organic carbon.
Right. Cool. Novak.
Yeah, I have a question. Help me understand this, or correct me in my thinking. So, based on the metabolism or the rates of metabolism that you see, these some of these organisms buried there have very, very slow metabolisms, and you're saying perhaps individual cell might live for hundreds or thousands of years based on the rate of metabolism. To me, that sort of sets up a contradiction, that if there's enough radioactivity down there to provide this hydrogen for energy, that radioactivity would be damaging the DNA that's there. And with the slow metabolism, these organisms would be inadequate to repair themselves. So can you somehow resolve that apparent contradiction where the radioactivity provides the energy, but there's not enough energy to have rapid growth to survive the attack or the disruption of radioactivity on the genome? Help me understand that.
I think this is a great question. And the first thing I'm going to say is that, you know, like when you have 1,000 cells in a cubic centimeter of sediment, and the average cell is a micron in diameter, a very small fraction of that total volume is actually microbial cells. So the odds of getting hit directly by alpha radiation are really low if you're living in that environment. That's the first thing I'm going to say. The second thing I'm going to say is my best guess is if a microbe's hit directly by alpha radiation, it's toast. Or probably more accurately, just splatter. But, you know, so like I'm sure you know that like the average alpha path is something in the ballpark of 20 microns. The average cell down there is about a micron in diameter. So the chances of a cell getting hit by any given alpha radiation event are pretty small. Over time, of course, you know, the chances of getting hit go up and up and up, but you're still talking about a very tiny volume of cells in a relatively large volume. So the statistics are in their favor for quite a while. The second thing I want to say is that the results are experimental and you know, observational. And so you're asking a really good question, and I don't know what happens to these things in terms of, we know that they've got genes that are active for reactive oxygen species, for managing those. You saw in the transcriptome that they're expressing a lot of genes for managing ROS. A lot, if they're not hit directly by the alpha radiation, they've still got to deal with the ROS from the alpha radiation. My best guess is that's why they're expressing those genes. They're trying to manage their reactive oxygen burden. The last thing I'll say on this is that we have sort of independent evidence for the long lifetimes and the slow biomass production, if you will. With Danish collaborators, we've done amino acid age dating of the biomass and the necromass in some of these sites. And those give us numbers that are consistent with the calculations based on the organic carbon energy fluxes. The amino acid ages calculated for the cells are in the ballpark of hundreds to thousands of years.
Wow.
And like, you know, I mean, it's one of those things where you're all aware that figures don't lie, but liars figure, right? So you know, you can poke at the paper and say, okay, where is there a loose link in this argument? And you know, then pry at it a bit. But you know, at the level that the calculations are done, it's internally consistent.
Okay.
Hey, Lisa, follow up on your chat. You wanted to get a reference?
Yeah, I would love to, maybe some names or something. I can look it up, but that would be great.
So I'm gonna type the name of the first author. I just paste the reference in here, but I'm afraid I'll kill the connection somehow.
Yeah.
And so if you look up Bente Lomstein and my name, I'm like the fourth or fifth author. Then you'll find it.
Okay.
Thank you.
And I'm sorry, that shouldn't be a quotation mark. That should just be an apostrophe.
Okay. Okay. So I think we might want to close off for today, but is there anybody that has a pressing question? I have some questions. Yeah, go ahead.
So my wife's a microbiologist, and so I like to think about bacteria and stuff. And I know one of the challenges.
Okay. Is that come on right?
They're trying to have balanced kids. Trying to I'm forgetting the word, to cultivate bacteria in a lab is very challenging for a lot of species. And so, but you guys have been or your collaborators have been growing some of these bacteria in a lab. Is that correct?
Yeah. So one of our collaborators, Heribert Sipionka in the early aughts, isolated a couple hundred different strains from seafloor sediment. About a quarter of them are basically the same thing over and over again, Radiobacter, a Radiobacter species. And then some of them are more exotic.
Yeah, just I know that that's a big problem with if you're trying to sample the things out of the soil, you can only kind of get a very small percentage of them to actually grow in the lab. And I just wonder, being under the seafloor, how do you, the pressures, obviously the nutrients, and it just feels like it would be hard to kind of replicate some of that environment. And if that's led to some, could that skew the results towards certain bacteria because those are the ones you're...
Yeah, we're out here right now to test something you just touched on. This particular expedition, we're coring in the Puerto Rico Trench. 2 or 3 days ago, we took the deepest water cores ever taken in the Atlantic. And we did that because we're trying to understand how pressure affects these populations and these microbes.
Yeah.
And one lab over, there are several microbiologists, Doug Bartlett and Ying Zhang and Bill Orsi and Doug's lab. And Doug and Bill are focused on doing experimental work to see how the community, how the organisms' activities and genomes respond to pressure.
Nice.
But you're right, it's a tough thing. And most of what we recover has not been cultivated. It's roughly the same categories of things are recovered from sediment all over the world ocean, and most of them have not been cultivated. Radiolytic, who I cited earlier in this part of our conversation, is fond of saying that cultivability is not a property of a microbe, but of a microbiologist.
Yeah.
And every time I quote him, I irritate every microbiologist in the audience. But Radiolytic grows microbes for a living, and his point is that we just need to think a bit and try harder. And that's a little glib, because probably not everything can be grown. But the people who are here right now are thinking and trying. And
that's really cool. I'll be excited to hear what comes out of that. That'll be very interesting. Thank you.
Thank you.
Yeah, thank you very much. This is very exciting. The questions could go on, but we have to do other things. But thank you so very much.
Thank you very much for inviting me.
Yes, accepting our invitation. And this will go down as one of the unique seminars off the coast of Puerto Rico.
That was awesome.
Yes.
On an expedition vessel.
Every time we send our robot down to sample the water, we have our Puerto Rican collaborators attach a Puerto Rican flag.
Nice. Take care.
Thank you very much for inviting me. Be safe in the snow.
All right, thanks.
There is more.
All right, bye-bye.